Very warm good morning, my dear students, and most welcome in the chemistry class on the chapter Surface Chemistry. Many topics we have completed. One is the adsorption we have completed. Catalyst we have completed. Enzyme catalyzed, that is we have completed. And today we present with the topic colloids. My dear children, you know that colloids chapter we have colloid topics we have completed. And this is the revision class on the chapter Surface Chemistry. My dear children, we have discussed that is colloids. It is a heterogeneous system in which one substance is dispersed as very fine particles in another substance called dispersion medium. In a colloid, the dispersed phase may consist of particles of a single macromolecules such as protein or synthetic polymers or an aggregate of many atoms, ions or molecules. Colloidal particles are larger than simple molecules but small enough to remain suspended. So the size of colloids are intermediate between the true solution particles and the suspension particles. Now students, what we have to know on the topic colloids? Colloids classified on the basis of the following criteria. Number 1, physical state, you know. Number 2, nature of interaction between dispersed phase and the dispersion medium, that is the lyophobic and lyophilic sol. And third type of particles of the dispersed phase, that is micromolecular colloids, macromolecular colloids and associated colloid students. Classification based on nature of interaction between dispersed phase and the dispersion medium. One is the lyophilic colloid students. What is lyophilic colloids? The colloids where the dispersed phase and dispersion medium, there is the less attraction between them and the dispersed phase is header of the dispersion medium, then it is known as the lyophobic. But lyophilic colloids means there is the strong attraction between the dispersed phase and the dispersion medium. The example is gum with a suitable liquid. An important characteristic of this salts is that if the dispersion medium is separated from the dispersed phase, the salt can be reconstituted by simple remixing with the dispersion medium. That is why these salts are so called the reversible salt. So, students, you have learned the difference between the lyophobic colloids and lyophilic colloids. Lyophilic, that is the dispersion medium header, lyophobic and lyophilic dispersion medium lover. So, lyophobic salts means where the dispersed phase and the dispersion medium, there is less attraction, and lyophilic colloids means there is a strong attraction between the dispersed phase and the dispersion medium. And lyophilic colloids examples, that is gum, gelatin, starch, etc. Next students, lyophobic, I told you, that is lyophobic colloids means the substance like metals. The word lyophobic means liquid heating. Liquid means dispersion medium. Substances like metals, they are sulfides, that is sulfur salt, gold salt. These are examples of the lyophobic colloids. Their colloid salts can be prepared only by special methods. Such colloids are called lyophobic salts. These salts are readily precipitated on the addition of small amount of electrolyte by heating or by shaking, and hence are not stable. So when you will differentiate the lyophobic and lyophilic colloids, first of all their definition must be written, the first point. Second point, it is not stable colloids but lyophilic is stable colloids. Lyophilic colloids is reversible but lyophobic colloids is irreversible. So these are the difference between the lyophobic and lyophilic colloids. Now students, already we have discussed the classification of colloids, multimolecular colloids, macromolecular colloids and associative colloids. Multi means many. Multimolecular colloids, on dissolution, a large number of atoms or smaller molecules of a substance aggregates together to form species having size in the colloidal range, that is 1 to 1000 nanometer. Students, can you tell me 1 nanometer equal to dash meter? You have to fill the dash. The answer is 10 to the power minus 9 meter. So, multimolecular colloids will be formed by the aggregation of large number of atoms or smaller molecules aggregations. This forms the colloidal size substances. The species thus formed are called multimolecular colloids. Multimolecular means many small atoms or molecules aggregate and form the species having the size of the colloidal range like as 1 to 1000 nanometer. For example, a gold sol may contain particles of various sizes having many atoms. Sulfur sol, gold sol. Generally, lipophobic colloids is the multimolecular colloids. Now students, you know macromoleculars. Macromolecules in suitable solvents form solutions in which the size of the macromolecules may be in the colloidal range. That is, the macromolecules in the suitable solvents form solution in which the size of the macromolecules may be in the colloidal range. Macromolecules, that is dispersed phase, when it dissolves in the dispersion medium, it forms the macromolecular colloids. Second, such systems are called macromolecules. Why? These colloids are quite stable and resembles true solution in many respects. Examples of naturally occurring macromolecules, students. Tell me, students, what are the examples of macromolecular colloids? Yes, your answer is absolutely correct. That is starch, cellulose, proteins, enzymes, and those of man-made macromolecules are nylon, polystyrene, synthetic rubber, etc. These are the examples of the macromolecular colloids. Now students, associated colloids. Associated colloid means there are some substances which at low concentration behave as normal strong electrolyte but at higher concentration exhibit colloidal behavior due to the formation of the aggregates. The aggregated particles thus formed are called micelles. In the exam, maximum times we have seen the question is coming, what is micelles? Actually, at the low concentration, some substances behave normal strong electrolytes, but at higher concentration, what is happening here? They exhibit the colloidal behavior due to the formation of aggregates, and the aggregated particles, students, are known as the micelles. And in this topic, two questions, maximum time coming, that is, what is Kraft temperature and what is critical micelle concentration. The two definitions are written here. The formation of micelles takes place only above a particular temperature called Kraft temperature, and above a particular concentration the micelle formation takes place, and this concentration is known as the critical micelle concentration. So what is micelles? What is Kraft temperature? What is critical micelle concentration? Students, this is very, very important. We have to learn and write this. Mechanism of micelle formation. Let us take the example of soap solutions. Soap is sodium or potassium salt of higher fatty acid and may be represented as RCO minus Na plus. As for example, sodium stearate, which is a major component of many bar soaps. When dissolved in water, it dissociates into RCO minus and Na plus, that is sodium ion, and RCO minus. The RCO minus ions, however, consist of two parts, a long hydrocarbon chain R, also called non-polar tail, which is hydrophobic water repelling, and polar group COO minus, also called the polar ionic head, which is hydrophilic part. So alkyl part R is the alkyl part. This is the hydrophobic part, and COO minus, this is the hydrophilic part. It is the polar part. As students, you know, like dissolve like. So polar part dissolve in water and non-polar part that is not dissolved in water. So in a soap there are two parts, polar parts and non-polar parts. One is the tail, that is the alkyl chain, hydrocarbon chain. Another is head, that is COO minus. It is called the head and hydrophilic part, that is water loving. Just to see here, soap molecules like as Na plus and RCO minus. Long hydrocarbon part, that is water repelling part, and short ionic part, that is water attracting part. Water attracting part. This is a soap molecules, my dear children. The RCO minus ions are therefore present on the surface with their CO minus groups in water, and the hydrocarbon chain are staying away from it and remains at the surface. But at the critical micelle concentration, the anions are pulled into the bulk of the solution and aggregate to form a spherical shape with their hydrocarbon chain pointing towards the center of the sphere. An aggregate thus formed is known as the ionic micelles. These micelles may contain as many as 100 such ions. Similarly, in case of detergent, as for example sodium lauryl sulfate, that is CH3CH2 whole 11SO4 minus Na plus. Students, you know that soap is the sodium potassium salt of higher fatty acid, and it is the sodium and potassium salt, but it is the sulfate salt, sodium or potassium salt of the higher fatty acid, that is known as the soap, and detergent is the sulfate salt. The polar group is SO4 minus, whereas the polar group in soap is the COO minus, along with the long hydrocarbon chain. Hence, the mechanism of micelle formation here also is same as that of the soap. So what is detergent? It is the sulfate salt and sodium or potassium salt of a higher fatty acid. So students, we have to learn the acids, that is the soap and detergent, and one question is coming, that is the cleansing action of the soap. Cleansing action of the soap. And this is the ionic micelles formation, and this is the stearate ion, water, arrangement of stearate ions on the surface of water at low concentration of soap, arrangement of stearate ions besides the bulk of water at the critical micelle concentration of the soap. Critical micelle concentration of the soap. So students, what we have learned? We have learned here the colloids, the definition, classification of colloids on the basis of the attraction between the dispersed phase and the dispersion medium, and the classification of colloids on the basis of the particle size of the dispersed phase. And we have learned here about the different types of colloids, that is macromolecular colloids, multimolecular colloids, and associated colloids. And associated colloids, example soap and detergent, and soap and detergents, that is students, we have learnt about the soap molecules and mechanism of the micelle formation, very very important parts we have learnt here. Next, what is soap? It is the higher fatty acids, salt, sodium salt of the higher fatty acid, that is sodium stearate, sodium oleate, sodium permeate. These are the examples of the soap. And the detergent, in case of detergent, there are different type of detergents. Ionic detergents, that is anionic detergents, cationic detergent, and the neutral detergents. We have already discussed these three type detergents we have to study. And cleansing action of the soap, that is the hydrophobic part and the hydrophilic part in the soap, polar part in the soap, polar part in the detergent, non-polar part of the soap, that is hydrocarbon, and non-polar part of the detergent, that is hydrocarbon part. And this is the cleansing action of the soap. And so students, we have discussed already, and this is the topic, subtopics we have completed now, and this is the revision class of the colloids, and already we have discussed it. Today again I have discussed, and I think 100% students understood the topic colloids. Rest next day we'll discuss. Today up to this. Thank you students, and be safe and be healthy. Okay, thank you.